Hey guys, so welcome to AP Biology, Unit 4A. As usual, just giving you a couple moments for y'all to get logged in. So don't forget, follow the absolute recap for, of course, some good podcasts in terms of AP Bio, AP Chemistry, AP Physics, AP Music Theory, and then, of course, I'm drawing a blank of their other one, but also follow Marker Learning for other AP needs. They just picked up a whole bunch of other AP subjects. So tonight we're going to talk about a couple different things. We're going to talk about cell cycle. We'll talk about mitosis, meiosis, and DNA replication. And so, as I said last time, put your questions down in the box, but a lot of times students aren't asking a lot of questions. So I figured that it's a lot easier for me to go ahead and have some type of visuals, because with biology, you really need some visuals. So I have some visuals for you guys, so we're going to kind of just go ahead and get started. And again, if you find that you have any questions about any of this stuff, just put your question down in the little question box, and I will, of course, answer it when I get to it, okay? So let's go ahead and get started looking at the cell cycle. So the cell cycle is pretty much the life of the cell, okay? And so as you see, we kind of start right here where we're going to go through our G1, then we'll come over here to the S, G2, and then we go through M. And at the end of the M phase, is when we'll end up having, of course, two cells, and those cells could continue going through this division, okay? So there's a lot of things that happen in this cell cycle, okay? So the cell cycle is made up of three main parts. You have interphase, which is kind of when it's preparing to get into that dividing state. You have mitosis, which is when we have nuclear division, and then you're going to have cytokinesis, which is where we're going to have cytoplasmic division. Yes, the cell can go through nuclear division without going through cytoplasmic division. An example of a cell that does this would be a muscle cell. A muscle cell is multinucleated, so you're going to find there's multiple nuclei within the cell because of the fact that they're so long and they don't actually go through a lot of division. So as an example of a cell that goes through mitosis without going through cytokinesis. So when we look at the cell cycle, there's a couple different things that happen. So there's a G0 state. G0 is a non-dividing state. This basically just means that the cell is not dividing. Okay, it hasn't received the signal to divide, so it's just kind of like hanging out, not dividing. Examples of cells that do this would be your nerve cells and your muscle cells, and then also your liver cells don't divide unless they get, of course, a signal that says, hey, time to divide, get that growth factor, and let's go. Which is why polio is such a problem, because it affects your nervous tissue. And so if you have damaged the nervous tissue from the virus, you're not going to be able to regenerate those cells because they don't go through cell division. So going back into interphase, so G1, G1 is going to be gap 1. This is actually when the cell is going to grow. It's going to get itself a little bit bigger. And so by getting bigger, it's going to allow for there to be enough space for when it divides, the cell will be the exact same size that it was before. We'll then go through the S phase, which is where we're going to go through the DNA replication. We'll make a copy of all the DNA. And then the cell also continues to go through this phase, but people kind of ignore that component. And then we also have G2, which is our second gap, which will be, again, the cells growing and dividing. Traditionally, you make copies of your organelles during the G2, but I wouldn't really focus too much on the details. The next part that we have would, of course, be the mitotic phase. Mitotic phase is just talking about either mitosis or meiosis, the actual act of the nucleus dividing, okay? And then, of course, last but not least, cytokinesis is when we divide the actual cytoplasm, okay? So as we said before, we have all of this is interphase, G1, S, G2, and then we have the mitotic phase, which takes up mitosis plus cytokinesis. And so it's important for us to recognize what's occurring before versus after mitosis or meiosis. So interphase happens before mitosis, cytokinesis happens after mitosis. So the next thing we need to look at is the checkpoints. Okay, and again, if you have questions, pop them down into the little question box. So with our checkpoints, the whole point of this was to make sure that our cell's division is controlled. Okay, you don't want your cells to be able to just continuously grow and divide, because that's what leads into tumors. So we need to have a way to regulate this, and we need to have a way to ensure that the cells are dividing at the appropriate rate and that they're dividing when they should be dividing. So we have a couple different checkpoints that are very important for us to know. Okay, so the first checkpoint that's really important to know is right here where we have our G1 checkpoint. Okay, so the G1 checkpoint is literally just saying, should the cell divide? Traditionally, there's going to be some type of growth factor. Okay, and so if it has that growth factor, GF is growth factor if you couldn't figure that out, which is going to be an external signal that's going to bind to the membrane, and through a signal transduction pathway is going to say, hey, yep, go ahead and divide. And it's going to get the signal to get past that G1 checkpoint, and so it will go and it will divide. Okay, so our next checkpoint that we have is our G2 checkpoint. Okay, at the G2 checkpoint, this is to say, okay, we have replicated our DNA. The DNA has been replicated. There are no errors that we know about, and we're ready to go through that division process. Okay, this deals with something called MPF. Maturation promoting factor. Some people just call it the mitosis promoting factor, but what it looks at is something called cyclin, and MPF is a cyclin dependent kinase. And if you can remember back to cell communication, kinase is a enzyme that's going to add phosphate, is in charge of phosphorylating something. And so as you go through G1, S, and G2, the level of your cyclin increases. So you have an increase in the cyclin level until it reaches its threshold, and that's when the cyclin and the MPF kind of bind together and the MPF starts working. And that allows you to get past the G2 checkpoint. And so at this state, we're just saying, okay, the cell has actually replicated its DNA. The DNA is relatively correct, and we're ready to go into the actual process of mitosis. So, and I have that right there. G2 is our DNA synthesize, MPF, maturation promoting factor, which is a cyclin dependent kinase. And then of course it looks at your cyclin levels. And I have seen before where there's like a diagram where we could look at the cyclin level increasing, and you have to figure out what part of the cell cycle you're in based on the cyclin level. Okay, so our last checkpoint that we need to talk about is our M checkpoint right here. Okay, notice that it's in the middle of the M phase. And so we know that the M phase is the mitotic phase, right? And so mitosis. So the M checkpoint is actually occurring during metaphase. If you remember that in mitosis, which we're going to go through in a second, metaphase has to do with when our sister chromatids or homologous chromosomes are aligned on the metaphase plate, if we're talking about meiosis. And so you're going to have microtubules coming from both of the poles and attaching to your sister chromatids or attaching to homologs, okay? If all of those sister chromatids are not attached, you shouldn't go through the process of anaphase, because of the fact that then you're not going to be equally able to split your chromatids or equally able to split your homologous chromosomes. So the M checkpoint is to ensure that we don't go through non-disjunction. It's to ensure that all of the sister chromatids or all of the homologs are attached to microtubules, so when we go through anaphase, we will equally divide the cell. Okay, so that's the point of the M checkpoint. So again, the G1 was saying, yes, we should divide. G2 is looking at, yes, we've replicated the cell's appropriate size. I've got the high level of the cyclin to allow me to get past this checkpoint. And then the M checkpoint was, again, looking at, do we have our microtubules attached to our homologs? Okay. Now, again, if you have questions, put them down in the little question box. I'm happy to help you guys and answer your questions as we go. I'm just rolling with what I have until you guys ask questions. Just in case, that way it's a little easier. Now, all the images I'm getting are from Creative Commons. So because of the fact that I'm making this live, I can't really use copyright materials. So some of my images are not the prettiest, which is about to happen with my mitosis image, because I couldn't find a good image that was not copyrighted. So sorry about this. So there are multiple phases to mitosis, and I already did the little mitosis dance, so you guys should be good on that. So again, before mitosis, we go through interphase. This is when we're going to basically make the copies of the DNA. The cell grows, as we just talked about. Okay, we move into prophase. The whole point of prophase is that we're going to prepare to divide. That P in prophase tells us prepare to divide. Our sister chromatids are going to condense down and they're going to find one another. Okay, you also see that we've got these spindles kind of moving to the opposite poles so that they'll be in the different sides, and you have your microtubules starting to kind of come out from them so that we can start to get ready. We're going to dissolve our nuclear envelope to kind of get ready to go into the next part. Okay, prometaphase is kind of an intermediate step. That's where we have our microtubules attached to our sister chromatids, and they're kind of having that tug of war to get them to line up on the metaphase plate. So it's more of a transitional period. Metaphase is going to be when our sister chromatids line up on that metaphase plate. They'll line up in the middle of the cell. Okay, and as you notice, we do have these microtubules attaching from both sides. Remember, this is when that checkpoint is going to be, and because of the fact that we want to make sure that everything is attached before we move past this phase. Anaphase has to do with the fact that now we have pulled those sister chromatids apart. We're going to break that bond that is going on in the centromere that's holding the two sister chromatids together. We're going to break that protein. It's a cohesin protein. So we'll break that, which will then allow my sister chromatids to move apart from one another. They move into opposite poles. And then the next part is telophase, where I'm going to form my new nuclei around my divided nucleus. And then cytokinesis is actually going to split. We'll have, since this is an animal cell, we're looking at actin, and actin is just going to basically create a cleavage furrow, which then separates those two cells. Okay, and again, that was mitosis. Now, the actual steps of mitosis is below the level of AP. It's considered just kind of a prerequisite knowledge. So what they're going to do is they're not just going to ask you a straight, which phase are you going to find the sister chromatids aligned on the metaphase plate, right? They're going to ask you an advanced question. They're going to say that there's a chemical, a cancer drug, and this cancer drug, let's say it's Taxol, is going to inhibit the microtubules. So they're going to say, which phase of mitosis will the cell be stopped at? And you think to yourself and say, okay, well, I know that in order for the microtubule to work, that's looking at anaphase, and that's pulling them apart. And if my microtubule doesn't work, then I'm going to be stopped in the phase before they are brought apart, which would be metaphase. Okay, so again, it's not a straight question. They're going to be asking you an application questions about these phases. So you have to understand the phases to be able to apply that knowledge. Okay, so again, if you have questions, pop them down in the box. But we're moving to our next part. Oh, I have an image for cytokinesis. So cytokinesis is different between animal cells versus plant cells. So as I said before, we're going to have this cleavage furrow. Basically, it's an actin ring that's going to pull really, really tight. And when it pulls really tight, what's going to happen is it just kind of pops them into two, because this is just the plasma membrane. And if you remember, the plasma membrane is made up of phospholipids, which are just independent little molecules that are free floating together. And so when we go and pull the actin close together, it just pops off and becomes two. So it's kind of think about like a bubble. You can make two bubbles from one bubble if you pulled it close and got the outside of the bubble together. And then over here, we see a plant cell. So with a plant cell, they're going to release these little, the Golgi bodies are going to have the secretions of the cell plate. And then we end up making a cell plate, which then, of course, ends up becoming our cell wall, because of the fact that we need to, of course, divide cytoplasm, which means building a new cell wall. So, again, if you have questions, pop them in the box. The next thing we need to talk about would be, of course, meiosis. Okay, so mitosis, we were making identical cells. We made two identical cells, in case I didn't say that. And so in this process with mitosis, we went and we replicated our DNA once, and we divided the DNA once. So we're able to make two cells, identical DNA. They started out as diploid, they ended as diploid, so everything stays the same. Okay. Now in meiosis, we're going to make 4 daughter cells. Those daughter cells are going to end up being haploid, and they're going to be unique. And so there's a couple things that happen during the process of meiosis that allows them to be unique. Okay. So first off, of course, in meiosis, okay, we're going to go through interphase, okay? So interphase, again, the DNA is going to condense into the chromosomes. So you're going to kind of see the little drawing there that they kind of did against down. Now, we're going through division twice, okay? So the first time we have prophase 1. Prophase 1 is, again, we're preparing to divide, okay? The same thing is happening in these phases that happened when we looked at mitosis. We prepare, we line up in the middle, we pull apart, we make a new nuclei, okay? The same thing is happening. The focus needs to be on what's different about meiosis. How does meiosis differ, and how do those differences allow for genetic diversity, okay? So in prophase, we already said we're condensing down, right? A difference is that here we're looking at homologous chromosomes, okay? And so just as a reminder, homologous chromosomes mean that we're looking at chromosome 1 from maternal and chromosome 1 from paternal, okay? So these are chromosomes that are the exact same length. They code for the same genes. So when I say it's coding for the same gene, I'm talking about it codes for eye color or hair color or height, the seed color, stuff like that. They're coding for the same thing. The alleles that could be different on them could code for yellow pod or green pod or brown eyes or blue eyes or whatever the actual specific allele is, which would just be differences in that gene. Okay. So as I said before, prophase is when we're going to have the homologs pairing up. Since our homologs pair up, we're going to be able to have something called crossing over. Okay, now I'm going to put you down because I need my hands for this. So with our crossing over, okay, we're going to have kind of our paternal, and then of course we would have our maternal chromosomes. Okay, as we said before, this would be our homologs. I probably should have drawn on them before I got started, but it happens like this. Okay, so of course, and I think you can see. So this would be our paternal, this would be our maternal, okay? And so of course, when we go into the S phase, we made a copy. So imagine my fingers are actually the same length. So these would all be chromosome 1. This would be paternal, these would be maternal, okay? So in crossing over, you're going to find that the non-sister chromatids that are on the inner part are going to overlap with one another. When that happens, they're going to exchange their genetic information, okay? So this blue one that I have is now going to have orange at the end, and this orange one is now going to have blue at the end. So when they cross over, now I have the orange on this finger and the blue on this finger, okay? So I hope that made sense. It's a little harder to do when you're virtual. I usually have my students switching shoes just to kind of help model it a little bit. So again, prophase is when we're going to see those homologs pairing up, and then we'll also see the crossover that occurs to allow for genetic diversity. Okay. So then we move into metaphase. Metaphase, the difference here is that now we have our homologs lining up on the metaphase plate. So when they're lined up on the metaphase plate, they're lined up in basically in the groupings of maternal and paternal because the whole homolog lines up. Now, when they line up, they go through something called independent assortment. Each chromosome independently assorts. What that means is that the chromosome 1 of paternal and maternal are independent for chromosome 1 as they are for chromosome 2 as they are for chromosome 3. Okay, so there's a 50-50 chance to see which side each chromosome is going to face. Okay, which means that since we have 23 chromosomes, there's 2 to the 23, 2 to the exponent of 23, different options of ways that they could line up for independent assortment, okay? Which, if I remember my math correctly, that's like 7-8 billion different combinations. But I can't remember off the top of my head. Anyways, it's 2 to the 23. So after we go through metaphase, of course, the homologs are what's lining up. Then we go into anaphase, which is where we're going to, this diagram is wrong. I should have noticed that earlier. It's the homologs that are split up. So ignore this little writing that's on there. So at anaphase 1, this is when our homologous chromosomes are pulled apart from each other. And so that's when we're actually going to have our cell become haploid. At the end of meiosis 1, your daughter cells are already haploid. Okay. Now, when they split, the whole sister chromatids go together. So maternal went one way, paternal went the other. So you still have a sister chromatid, which means you still have a duplicate, okay? Which is why we will not go through interphase in terms of replicating our DNA after we end with telophase 1. Okay, you're not going to need to replicate your DNA because you already have a replicate. Okay, the cell's going to need to grow a little bit. We need to make copies of some of the organelles and get bigger, but overall, you're not needing to make copies of DNA, so that's a lot faster of a second interphase, but it's not really an interphase. They call it something else. Don't worry about it though. So then we move into our second part of meiosis. So we are now going to go through prophase, prepare, sister chromosomes line up, metaphase, sister chromosomes line up on the metaphase plate, anaphase, sister chromosomes get pulled apart until we finally end up with 4 daughter cells. Okay, so meiosis 2 is exactly like mitosis. The only difference is that in meiosis, the chromosomes aren't identical because of the crossing over that happened in meiosis 1. So how many sets of chromosomes are present at the beginning of meiosis? Okay, at the beginning of meiosis, you're going to start out being diploid. Okay, and then what's going to end up happening is that you go through the first meiosis 1 and the cells will be haploid. And at the end of meiosis 2, the cells are still haploid. So I hope that answered your question. If not, please let me know. I can get you back in there and answer it again. So I think we've gotten that. Okay. So it was brought to my attention that I am not appropriately meeting your needs because I'm not asking you guys to do the types of questions that they're going to ask on the AP exam. So I pulled another free response question for you guys, so you would be able to see what they could ask you about this. Okay, so this is from the 2015 exam. So on this question, they're saying that both mitosis and meiosis are forms of cell division in which they can pull genetic information. So they want you to describe 2 events that are common between them. So when we think back to mitosis and meiosis, they both have interphase that happened before, right? So you could say that in interphase, we're going to make copies of DNA before. Check. Okay. We also saw that you're going to have something lining up on that metaphase plate, whether it is the sister chromosomes lining up on the metaphase plate or whether the homologs. In both processes, you're going to find some of our chromosomal material lining up on that metaphase plate so that we'll be able to equally separate that genetic information. Okay. We also saw that you're going to condense the DNA. I like to tell my students that it's like you have a plate of spaghetti, right? It's going to be really hard for you to separate that plate of spaghetti. But if we go and we take a fork and we wrap each noodle around a different fork, then you're going to easily be able to separate that spaghetti. Well, by condensing your DNA down and putting your DNA around a fork, okay, or around something called a histone, which is the protein that you use to organize the DNA, by wrapping it up and organizing it that you do in the prophase step, it's going to be a lot easier to separate it. And now both of them, of course, go through that process of condensing. Okay. So the next part that we have that could be similar between them is cytokinesis, right? Cytokinesis happens after mitosis, but you could still say that in both of these processes, you go through cytokinesis, okay? And I see that I have a question box. Is N the number of chromosome sets? Yes, N is saying the number of chromosomes. So if you're haploid, you're just a single N. If you're diploid, you're 2N. If you're tetraploid and you have 4 sets of chromosomes, then you would be 4N. So you're just looking at how many sets do you have. So we're considered diploid or 2N because you have 1 set that came from maternal side or your mother, biological mother, and 1 set that came from your biological father. So 1 set from sperm, 1 set from X. I hope that answered your question. So moving on. Again, if you have questions, I'm answering them as we go. So pop them down to the little question box. So the second part of the question asks for you to describe 2 features of cell division processes that lead to differences in the actual cells that we get in mitosis and meiosis. Well, let's think. The differences that we saw. Well, number 1, we go through 1 round of division in mitosis, but in meiosis, we go through 2 rounds of division. In mitosis, we're going to end up having 2 cells. In meiosis, we have 4 cells. In mitosis, your cells are going to be identical. In meiosis, your cells are going to be unique. Another difference, when we go through mitosis, you end up with diploid cells. Meiosis has haploid cells. In mitosis, you do not have crossover. In meiosis, you do have crossover. And then independent assortment. In mitosis, you do not have independent assortment, but in meiosis, you do have independent assortment. Notice that for every single one of those differences, I mentioned mitosis and I mentioned meiosis. A lot of times when they're wanting you to basically say how things are the same or different, you need to describe both of them. By saying the independent assortment occurs in meiosis does not tell me that it doesn't occur in mitosis. And the readers need you to show them you know what's going on. They're not going to assume anything, okay? So be very clear. Mitosis does not have an independent assortment. Meiosis does have an independent assortment. And of course, also, it says describe. So you would have to describe that phase. You can't just name drop things, okay? So again, if you have questions, pop them down there. But that was actually a question. It was the 2015 question, and it was number 4 if you want to go back and look at it. I am going to export this PDF, and I'm going to put it onto the page like I did last time. So if you want to go back to this at any point. But as you notice, most of them are just images. But I do have all that for you. And so if there's any question on mitosis and meiosis, pop it down to the little question box. And now we move into DNA replication. So this gives us a big picture of the DNA replication. Okay, so you can kind of see that within replication, we have 2 strands. There's a leading strand and a lagging strand. You also see there's different enzymes. You have the enzyme helicase. You have the enzyme DNA polymerase. There's a couple other enzymes that are not labeled here, like topoisomerase and primase. So it's important that we know those enzymes, okay? So first thing we kind of have to think about is DNA is anti-parallel, right? So you have one strand that is going to be 5' to 3' moving in this direction, and you have one strand that's moving 5' to 3' and it's in this direction. So they are equidistant apart, if you remember, because of the fact they each have one pyrimidine and one purine. As a reminder, your purines would be your adenine and your guanine. The way that I try to help to remember, and again, this is just a mnemonic device, is that if you're from Georgia, GA, you're pure, the purines, GA. And then, of course, the pyrimidines would be cytosine, uracil, and thymine. So you want to cut the pyramid. So CUT, cytosine, uracil, thymine are the pyrimidines, and then adenine and guanine are your purines. And as a reminder, the pyrimidines have the single ring structure, and the purines have the double ring structure. And so when we pair up a purine and a pyrimidine, you have 3 rings going across your helix, or making the width of the helix, so they would be equidistant. So, back to what I was saying. So they're anti-parallel, so they're going to move in opposite directions. Your 5' end has a phosphate group, while your 3' end has a hydroxyl group, okay? When you replicate DNA, it always moves in the 5' to 3' direction. Let me say that one more time, make sure you've got that, okay? When you replicate DNA, it always moves 5' to 3'. That means that it reads it 3 to 5 because they're anti-parallel, okay? When you pull the strand apart, one of those strands will already be set up in a 3' to 5' direction to be a template, while the other strand is going to be moving in the opposite direction, okay? So that's why we have a leading strand and a lagging strand. The leading strand moves continuously toward the replication fork. As the strand opens up and as you separate them by breaking those hydrogen bonds, okay, one of your templates is already in the correct direction, so I can just move directly down that template, reading and going through complementary base pairing. So as I continue to open up the DNA, then I'm going to be able to continuously move toward that replication fork. That is your leading strand. Your lagging strand is going to move in opposite direction, okay? You open up a little bit, lagging moves backwards. You open up a little bit more, lagging moves backwards. You open up a little bit more, lagging moves backwards. You continuously have to jump back and keep going forward again, okay? So that's the big difference between the leading strand and the lagging strand, okay? So as I said before, the leading strand is going to move continuously toward the replication fork, while the lagging strand is going to be moving discontinuously. We're going to make a strand here. We'll jump back, make another strand. We're going to keep having to go backwards and making new strands backwards. So there's a couple different enzymes we need to make sure we know. First enzyme that's important is helicase. The job of helicase is to break the hydrogen bonds. The job of helicase is to split and basically break the bonds between those nucleotides so we're able to make the replication fork. So we're able to separate those two strands from each other so that the strands can be the template for the next strand. We call this a semi-conservative model. The next enzyme that's important is something called primase. Primase is going to make an RNA primer. Okay, so that means that we're going to put RNA nucleotides in. Okay, so uracil will be in there. It's going to have ribose instead of deoxyribose. And this creates about a 10 to 15 nucleotide primer. Why do you need a primer? Because DNA polymerase can't build off of nothing. It needs to have an open 5' end in order to, I lied. It needs an open 3' end in order to build off of it to keep going, okay? So unless it has a starting point, DNA polymerase can't move, okay? And so what that means for you is that we need to have that primase, okay? So our discontinuous is going to keep having to use primase and it's keep jumping back, and we're going to have to keep replacing all those RNA nucleotides. DNA polymerase, it of course tells you what it does. It makes a DNA polymer, so it's the one that's responsible for actually going through the base pairing. You're going to notice in your textbook there's a DNA polymerase 1 and DNA polymerase 3. It is not important for you to know the difference between those. Just do know that DNA polymerase is going to be what makes the copy, as well as it's going to be what replaces the RNA nucleotides with DNA nucleotides. And last thing is it's going to do the erroring. So if there's been an error made, so instead of adenine binding with thymine, there was, I don't know, a cytosine that bonded with thymine. That's not logical. A guanine that bonded with thymine. It'll pull out that guanine and it will replace in an adenine. So we did that one. And the last enzyme is topoisomerase. Topoisomerase's job is to relieve the strain. So as you unravel the DNA, it kind of gets super coiled upstream. So it's going to basically break the bond, unravel a little bit, and then rebond. So that was our quick little review over mitosis, meiosis, and DNA replication, and of course the cell cycle with its checkpoints. Do we have any questions before I log off? Remember, don't forget to subscribe to the Absolute Recap and Marco Learning because they have a lot of great resources in your other AP Sciences that might be helpful. And I'm not seeing anybody posting any questions, so I hope that you guys have a wonderful day. And remember, you're AP Bio Penguins and you're dressed for success. Bye.